My name is Blanche McCormick. I serve as director.
And welcome everyone. And we're going to start right off with our first speaker for the FYRE and SURF Undergraduate Summer Research Showcase. And Kyla Schmidt is our first speaker. So Kyla, you should have control.
Is my slideshow showing? Okay, perfect. So this summer, I opted to do research in Tran Creek, which is a local creek near me, which I'll get a little bit more into later. But I did research on crayfish. Specifically, I did monitoring across 3 months. And it was a community research program. And so that meant that not only was I researching, but I also was able to bring in the help of friends, family, and just organic community volunteers to help me with this kind of data collection in the field. Okay, a little bit of background is that Tran Creek is a nearly 8-kilometer waterway that receives 9 smaller creeks. It's a pretty significant cold water habitat in the Portland and Lake Oswego area because it provides refuge for a lot of the species that need cold water from the Willamette, which can get warmer during warm months. And so its placement makes for crucial habitat, and also it's a good research setting because at multiple points throughout its length, it's surrounded by publicly owned land, which makes for a simpler access when you're looking to collect data. And so that was kind of a factor in when I was choosing my research site. And then onto the species. So Tran Creek is home to a species of crayfish called the signal crayfish. And it's one of Oregon's, or it is Oregon's only native crayfish species. Lots of states have up to 10 or more native species, but we just have the one. Here it's a keystone species because they provide really essential roles in cycling nutrients to the ecosystem, providing aquatic nutrients to terrestrial predators, and they also break down plant matter and dying animal flesh and all that stuff. So they're really good recyclers. They're also ecosystem engineers in that they can kind of affect how sediment gets distributed because they burrow into the riverbed and have different habits that can just affect that distribution of sediment. And so in those ways, they're very influential. And lastly, they're kind of an economic staple as well. The crayfish industry is not as big in Oregon as it is down south, but it does exist. And they also do provide essential food for other game fish that people like to catch, like bass and pike minnow. When I was starting this research, one of the main things that I was doing, aside from just general population characterization, is looking at potential problems that they may be facing. And so those could come in the form of non-native crayfish being introduced into the area by either science projects. I think a lot of us in school growing up had crayfish units where we studied crayfish. And if students take those crayfish home, they could release them into the wilds. And if they're not native, they can become introduced. Or if fishers like to use them as live bait, they can become introduced into areas where they're not supposed to be. And they can actually end up causing millions of dollars in damage, as well as helping to make extinct lots of native species. And so that's kind of a big threat. We have seen reports of them in the area. So I was looking to see if those existed in Tran Creek. They also can be threatened by human infrastructure. And as I delved into in my research, there's specifically a culvert that is kind of at an essential point that connects Tran Creek with the Willamette River. And it really is this key point as to preventing species from being able to navigate between the two. And so I was looking at how well crayfish are able to kind of deal with the presence of that human infrastructure. And then lastly, for crayfish, this is more of a natural phenomenon, but population density can really affect survivorship, as these organisms are pretty aggressive and can be competitive when there's not a lot of space, and can even result in things like cannibalism when there isn't adequate room for their population to grow. So just looking at those different factors that can occur. And then my research, I guess, it really began last summer when I had kind of inspiration from the Tran Creek Watershed Council to go ahead with this project that I had been thinking up, and they helped advise and implement that. And I actually was able to publish that research, which was really awesome. And it actually just finally went through the process like the other day, even though it was from last summer. And so that's really exciting. And so that research kind of helped me narrow and figure out what could I do better this year and what issues should I really be focusing in on. And that was kind of where my new peer mentor helped, as well as continued support from that council. And so how did I go about this? So the main thing that I did was nighttime crayfish surveys. Crayfish are nocturnal, and so I'd be going out at night to the creek. We considered using traps, but we just decided that we're going to get more, we're going to catch more if we go in by hand, because traps can only hold so many. And so I ended up actually doing 27 surveys throughout the summer. I had 9 sites, and I'd go to each site once per month across 3 months. And it was a catch and release process. And so we'd use dip nets to catch crayfish for a set amount of time, usually 25 minutes. And then at the end of that period, we would have placed them all in separate containers, and we would then proceed to check them for these mutualistic worms that can sometimes be found on crayfish. We'd measure them, we take pictures for iNaturalist, and we would look at other factors like coloration and whether they're missing different limbs, which can be a sign of like aggression and competition. And so then I also did just recently a behavioral kind of follow-up case study in the culvert, just to see more specifically how they interact with it, and was able to see a couple interesting interactions taking place. Also did some supplemental environmental data collection just to help characterize different crayfish population sizes with their environment. And so looking at things like flora richness along the banks of the creek that were being studied, and then water characteristics like pH, alkalinity, temperature, and how wide the stream was where we were surveying. And then in terms of kind of analyzing, corroborating, and storing the data, I would input a lot of my initial metrics into Excel/Google Sheets platforms. And there I was able to really use more mathematical models, simple ones, but just using them to estimate things like carapace length from total length and thus their age. On iNaturalist, it was a really cool platform. It allows you to upload photos of the species and help corroborate others' identifications. And so I had my peer mentor help me identify plants samples that I would upload. And I also had others help me identify crayfish and make sure that the ones that I were looking at truly were signal crayfish. And then finally, SPSS was helpful for running statistical tests and producing a few graphs. And so this is just a little collage I made of some of the volunteers that came out and helped me, and just highlighting the contributions that people made to my community research, and that made it a really special experience. And so finally, we come to the preliminary findings. It's still very much a work in progress. And so I haven't analyzed everything that I want to yet, but I've started playing around with the data. And I guess in terms of behavior, I found, and this is kind of reflected on the right there in those 2 pictures, that crayfish both had positive and negative interactions with human infrastructure. And so on the right, you can see there's actually a crayfish that's climbing up out of the water to go around a baffle in the culvert, which was placed there to help with fish passage, but can kind of interfere with other animals. And so you can see that in some regards, they have adapted to that environment. But on the left, you can see there's actually one that became wedged inside the baffle and was unable to escape. And so obviously we are seeing that human infrastructures are having some negative impacts as well on these populations. Statistically, I was able to determine that larger crayfish that we found were more likely to have these symbiotic worms living on them. And there's still research being done as to whether these worms are parasitic or actually mutualistic or something else altogether. But knowing that information is helpful when down the line we are able to figure that out. I also use an existing kind of model that other scientists had constructed to relate crayfish length with the age. And I found that the population I sampled from was relatively young, with a mean age of just 1.9 years old. Many crayfish live to 10 years. And so seeing a population this young could indicate that it's becoming too dense, and that higher competition is kind of not allowing them to live to the full length of their lifespans, and is decreasing that expectancy. And so with that, that's all of my findings so far. I look forward to being able to present further once I have analyzed more of that data. But I'd just like to conclude by saying that this was an amazing experience. I loved being able to get out in the field and work with friends and family and community members to sample these very interesting animals and learn more about them. And I'm really grateful to everyone who made this possible. So thank you.
Thank you, thank you, thank you. I'll put my clapping emoticon on. And congratulations on getting published. That is really, really wonderful. Congratulations. All right, we have our next speaker, Zoe Bailey.
All right, can you see my slides?
Yes.
Awesome. Okay, so this summer I was looking at the effects of burn history on the seed bank composition in the North Great Basin, and this work was made possible with the support of Dr. Lauren Hallett's lab. So just for some quick orientation, the Great Basin is the large region of land that stretches from the Sierra Nevadas and the Cascade Range all the way east to the Rocky Mountains. But my project was specifically looking at the part that extends up into Southeast Oregon, the North Great Basin. So a little more information about the North Great Basin. It's a semi-arid shrub steppe ecosystem. So there's a lot of this sagebrush, which you can see along the fence line in this picture here. And the sagebrush creates a really unique and special habitat for species of conservation value, such as the sage grouse. More specifically, I was working in the North Great Basin experimental range, which is marked here with this big red star. And it's a facility jointly operated by the USDA and Oregon State University. And because of a few connections through Dr. Hallett's lab, I was able to work there with an Oregon State fire ecologist, John Bates. A little more about the site. It opened in 1936 and was originally used for sheep grazing. So the sagebrush was cleared out. These days now there are cattle instead of sheep that are rotated around. And it's a unique site for an ecological study because there has been many historic projects there with different rotations of cattle, drought simulations, and also the application of fire. My study was specifically looking at seed banks, which many people would correctly first think of as the sort of preparation for apocalypse, storing seeds in a vault in Norway. But in this case, I'm thinking about seed banks as the accumulation of seeds in the soil, and I measure it as the combination of seed density, so the number of viable seeds that could germinate, as well as the species richness, so how many total species I found in a defined area. And this little figure on the right I'm borrowing because it really nicely demonstrates how some of the seeds will germinate, others will remain in the seed bank, and some won't germinate at all. And this process of whether or not they germinate is influenced by whether or not there's fire on the landscape. So fire can have a few really direct effects. The smoke and ash, as well as the heat shock, influence a seedling's ability to germinate. There are also indirect effects. Soil properties change after fire. This figure is showing how the depth of soil that repels water is extended after a burn. And fire, of course, removes all the above ground vegetation. So this really changes competition dynamics for light and water, and which species and which functional groups will do really well after a disturbance. This is an important type of disturbance to think about for the Great Basin in the changing climate that we're experiencing. So not a new story, but specifically to the basin, spring is now being observed about 10 to 15 days earlier than it was in the mid-1900s. And this projected warming trend is expected to continue. I just grabbed an estimate from the US Forest Service that within the next century, the area is expected to warm another 3.6 to 9 degrees Fahrenheit, which is a very significant change. And this could potentially increase the frequency of fire and the severity of fire in the landscape. So disturbances like fire tend to favor species of plants that can grow really quickly and take advantage of all the new resources that are opened after the fire. So species that can focus their resources on seed production instead of establishing deep roots initially tend to dominate disturbed landscapes. So these species are usually annual plants that reseed themselves every year. And because of that process, they have a unique ability to disperse many seeds at once. So here are a few of the species that I saw in my plots broken out into functional groups. So perennial species can persist year after year versus those annual species that have to reseed themselves every year. So the only annual in this figure is all the way on the far right. There's the annual grass, the invasive weed, cheatgrass, Bromus tectorum, and you can see those really shallow roots compared to the above ground biomass. So species like Bromus tectorum, aka cheatgrass, tend to invade after fire. And this annual invasive is a particular concern in the area. It was brought by European settlers in the mid-1800s, and the settlers at the time grazed the area very heavily. So the native perennials were pretty much cleaned out in certain areas, and it allowed for this invasive to become very established. It also germinates sooner than native grasses, germinates in the middle of winter, as opposed to the regular spring timeline for most natives. This gives it another advantage. And so now these days it's displaced about 70 million acres in the Great Basin. So it's pretty significant, in part because when it dominates, it displaces those native grasses, which are preferred forage for cattle. So it can affect the way of life for ranchers. It also poses a unique fire risk because cheatgrass grows really dense. It grows close to other cheatgrass plants, and this creates a continuous fuel base for fire to spread very quickly across the landscape. It also dries out earlier than native vegetation and grows with a lot of above ground biomass, so just lots of leafy shoots, and they are very thin and can ignite easily. So it creates this perfect storm for the potential for a positive feedback loop, where we have increasing fire risk from warming temperatures, increasing invasion of annual grasses after these fires that then make that landscape that much more vulnerable to burn again in the near future. So there's a lot of ongoing work to break this annual invasive grass invasion cycle that's being done in part by the manual seeding of native grasses by land managers and by the Bureau of Land Management. And this research attempts to contribute to that effort by asking a few questions that remain as gaps in our knowledge. So I asked how fire history affects the species richness. So again, the number of species and the species seed density in the seed bank. And I was also curious if there was a relationship between the fire history and the degree in which the area was invaded by that annual cheatgrass. So to answer these questions, I sampled in some unburned plots that had never received prescribed fire, as well as plots that have been burned by Oregon State every 5, 10, and 20 years. And I collected soil at 2 different depths to see if the dynamics of fire affected the soil closer to the surface as opposed to a little bit deeper down. And I rented a truck and rented some equipment and went out for a few weekends this summer, stayed on site, went with a friend from my lab, Ellie, who was kind enough to come during a heat wave. And we collected soil and identified plants in those sampling frames, as well as recorded cover data. So looking at what fraction each species made up in our sampling frames to extrapolate to the larger plot area. And then this is just a very preliminary figure. I'm still processing most of my data, but this is an interesting finding that I found from the field that's looking at the percent cover, so that fraction of my sampling frame that the cheatgrass made up. So the figure on the left, we've got the average percent cover of cheatgrass in the burned sites versus the unburned sites. Burned is the yellow bar there. You can see I found about 6% of each sampling frame was cheatgrass in the areas that were burned, as opposed to almost half that, 3% in the areas without fire. So pretty significant difference initially there. And then a similar finding on the figure on the right, still have cheatgrass percent cover on the y-axis, but this time I have the frequency of fire on the x-axis. And you can see that as fire frequency increases, the fraction of my sampling frame that the annual invasive made up also increased. So a few next steps, I'm still in the process of growing all the seeds that I collected in the soil. So I'm using a growth chamber and playing around with different watering regimes to see what my highest chance of survivorship is. So I don't have data from that yet, but that's what I'm working on this fall. And this fall, I'm also hoping to return to the same site and sample again, because the site that's burned every 5 years is up for its burn. So hopefully I can help with the burning process, but at a minimum, I'll go back to that transect and sample the soil immediately after the burn to see if the effects of fire change with time or if they're pretty similar immediately following fire. And then just a big thank you to all the people in the Hallett Lab. This photo on the left is just a fraction of the crew that's helped me this summer with mentorship and inspiration for this project. Special shout out to my advisor, Lena, who's here with the backpack in the forest, and to my PI, Lauren Hallett, and of course to Ellie, who came out to sample.
Thank you so much. For those of you doing field work, Kyla and Zoe, the weather this summer must be fascinating, right? And especially in continuing with your research and how it will continue to affect your research as well moving forward, right? Depending on how the climate will be in the next few weeks, few months. So thank you. All right, next up, we have Jenna, who's going to tell us about toys and words in everyday space.
Hello, can you see my slide?
Yes.
Awesome. So first, I just wanted to thank you, Kier, for giving me this space to share about my awesome summer research, and thank you all for being here today. I am super excited to talk to you about how an infant's environment relates to word learning. So my original research question asked, does the spatial organization of toys being placed in one area versus scattered throughout a home matter for how children learn the toys' names? So to understand this a little bit better, I think it's helpful to kind of dive into why an infant's environment is important. An infant's environment is important because it's where they feel the most comfortable and where they act the most authentically. So to understand the benefits of researchers in a home, I think it can be helpful to kind of contrast that with the opposite, which would be an infant study in a lab setting. So on the left, you'll see an image of a researcher playing a game with an infant. This is the traditional method to understand how infants interact with what's around them. And it's provided researchers with a great understanding of infancy development. But with recent technology, whether that be a camera that the infant is wearing, a researcher filming in your home, or even text messages to parents, researchers can get a different view and perspective on how an infant interacts with what's around them and how they can learn from that. So in a home environment, the child is comfortable, they're acting how they would on a day-to-day basis, whereas in a lab study, they would simultaneously need to be adjusting to their environment, so certain interactions might not occur as authentically as they would in a home. So to do this, to kind of dive into an infant's environment, I joined a research project called Play and Learning Across a Year. This is a nationwide project with researchers located all over the US. There are 4 components to this project, as you can see on my slide, which adds up to about 3 or 4 hours of data. The data collected needs to be uniform because researchers use it to answer varying questions. So we spend a lot of time practicing to ensure this uniformity. Above is a photo of myself and mentor Dr. Fozzie working on the questionnaires, and then also a picture of our undergraduate play team. I believe that day we were testing out the camera equipment. There are lots of parts and pieces to this project, and so we had to kind of get comfortable using it all. For my specific question of toys and their location in the home, I'm primarily interested in the house walkthrough portion as well as the questionnaire portion. So the house walkthrough provides a map of where the toys would be located in the home. And then the questionnaires provide an assessment of the infant's vocabulary. So this is an example of the word portion of the questionnaire. It uses the MacArthur Bates CDI to record how many words a child knows. These lists cater to both monolingual and bilingual infants. The lists were carefully crafted to ensure that the words are words that a child at that age should know or might know. And then based on which words they do or do not know, other inferences about their overall development can be made. However, for my particular question in my research, I'm simply using it for just a measure of total words the child knows. So then the house walkthrough portion is important because the way that the objects are arranged in a home can tell us a lot about how a child will learn words. Each situation shown on this slide provides both opportunities and challenges for learning. For example, in the example on the left, the piano would be a stationary object. It's predictable, it doesn't move, and it would be easy to associate the word piano with what it is, since it doesn't necessarily move. A child would associate piano with, in this image, under the books or next to the basket or even tucked in a corner. The challenge with this is whether or not the child would recognize a piano in another location other than the one that is familiar to them. So to contrast with that, the example on the right shows an object that is not stationary, but rather is moving throughout the home. In this case, I chose my object as a rubber duck. The object is always the same and may appear more frequently, which is an opportunity for word learning, but the environment changing would be a challenge because it makes it harder to learn the name of the object without associating it with something like a location. The important thing to remember with this type of data, and especially the play project, is it collects a variety of data, and it takes a lot of time to collect the data due to an entire family needing to dedicate a large amount of time toward the research. As well as having researchers in a home during a pandemic, which can get kind of challenging. So we have taken a pause on this project, and we will resume data collection when COVID numbers decrease. In the meantime, I had a lot of time in the lab to kind of learn some other components of research. So to work more efficiently and more effectively with data that we've collected, a lot of tools are necessary to take, like, for example, a raw video file from the play project and turn it into something more manageable and easier to look at. So data view is a really good example of this. It allows us to pinpoint certain locations within a video file and organize it based on what we're looking for. So for example, to answer my question of the relationship between toys and words, I may pinpoint when I saw a toy in a video or when I heard child speech. And then once those video files are organized, we can take that data and put it into R, a coding software that gives us the statistics we need to answer the questions. So like I mentioned, with Play on a Hiatus, gave me a lot of time to work with these tools. And then by the time we move on to the next steps within working with data, I'll be ready to go. So in fact, there was some data that we started working with. Ecological momentary assessment. So as I briefly mentioned, the play project takes a lot of time and a lot of resources. Object EMA, or ecological momentary assessment, answers similar questions, but in less time. This research is more cost effective and it makes it easier for parents to participate. So there's a larger rate of parent response. For this study, parents agreed to a few sessions in which their phone would ping throughout the day, and then they would be responsible for typing in what object their infant was holding. Parents were asked a series of questions relating to their child, so their position, are they sitting, laying down, the activity they were doing, and then whether or not they were holding an object. As you can see on the timeline, this was collected through 4 sessions. Each session lasts 4 days, and then there were about 10 notifications per day. So this object data can be used to make inferences about how often objects were held, similarities between objects, and then similarities between objects and time of day. We started with a sample of about 6,000, each entry looking something like the word cloud you see above. And one of my tasks this summer was to clean the data to make it consistent so that when we work with it later, it's a little bit easier. So for example, taking a look at the word cloud, a bottle of Pepto-Bismol to shake simply becomes bottle. And you, Gary Meltz, we decided was a typo or an autocorrect incident for yogurt melts. So with all of the work making this data consistent, you might be wondering why we don't just send off a list of objects to parents, but we could do that, but then we wouldn't get the variety or the accuracy of what the infants were necessarily holding. For example, in that if we were to make a curated list, I never would think to put grass and leaves or zany zoo on that list. So doing it this way was really beneficial. So where do we go from here? Summer was a time of learning and a time to kind of get a grasp on all of these up and coming projects. The blue histogram on the slide shows the comparison between a few objects from the object EMA data and how frequently they appear. Many common objects such as bottle and the generic word toy repeated many times throughout the 6,000 data entries. Others such as diaper and banana occurred less frequently, and then most words were unique. They occurred only once throughout the set. So over the next couple months, we'll be able to make more observations using that data. And then to further my understanding of spatial relationships and word learning, we plan to combine the projects. So a sample of infants participating in both the place study as well as object EMA, and that might just open the doors I need to answer my question. The possibilities are endless when a home video and a list of unique objects are combined together. We can compare the objects to the video as well as the MCDI score to determine overall language knowledge and then whether or not all of those pieces are connected. In the meantime, I'll be in the lab with all the other amazing undergrads and my mentors, Dr. Fozzie and Ali Kuznia, and also helping recruit participants for our studies through Team Duckling. So thank you so much for your time today. I hope you learned something about infant language and word learning.
Thank you, thank you. It will be really interesting once fall moves forward and you're able to make more home visits, right, and the environment changes, how that will add to the research. So thank you so much. All right, next up is Amelia, switching gears to streaming racial categories, Spotify, K-pop, and genre.
Sorry, can you see the PowerPoint? Oh, thank you. Hi, I'm Amelia Detweiler, and I'm grateful to be able to present today. My project, like Lance said, is streaming racial categories, Spotify, K-pop, and genre. My faculty mentor was Dr. Jina Kim, who is amazing and is actually doing research in Korea this summer as well. So I'll start with a brief explanation of what led me to this project in the first place. As a Korean American adoptee, I've become increasingly curious and concerned about race relations in the US. So here's just like a couple ways how anti-Asian hate has become more visible throughout the pandemic. There were over 10,000 reported hate crimes, and the Atlanta shooting on March 16th of last year, 6 of 8 women murdered in spas were Asian. So as I digested these things that were happening, I was also keeping up with pop culture and music because I'm interested in that. So relating race and pop culture, specifically music, the Grammys have a long history of racism or Black marginalization. Clearly seen through artists like The Weeknd and Beyonce either not being nominated or not being awarded. However, in 2021, K-pop peeked its head into the Grammys scene. BTS became the first Korean band to be nominated, and many expected them to win. But when the award went to 2 white women with a less popular song, many people looked to racism and xenophobia. So all of this together made me think about race and the way cultural products engage with race relations. And so I developed this project with Dr. Kim. existing scholarship is often concerned with the Korean wave as a phenomenon, like Squid Game, the recognizable things. Why did it happen? What is in the industry that makes it happen? Or what even is K-pop? But my goal was to engage K-pop and race relations together in relation to Western pop and outside of the black-white binary that is often discussed in music, with R&B and hip hop being like the primary units of analysis for race in music. So my first question was, what criteria does Spotify use to classify a song as K-pop? Are there musicological or lyrical distinctions between K-pop and Western pop? So basically, the question is whether there's actually a difference between pop and K-pop. My second question was how this categorization draws upon or complicates racial and ethnic relations. Because of this genre segregation, I wanted to engage music as an expression of race relations in like the quote real world. So for my sample, I chose to use Spotify because it's the largest streaming platform. And so it has a strong hand in shaping people's perceptions of genre. I had 525 songs from their editorial playlist. I chose a few playlists for each genre with the aim to provide a well-rounded variety of songs within the genres. And their release dates are graphed here. K-pop, nothing from the sample was before 2016. And that's due to it being a relatively newer music genre on the global scale. So with these songs, I decided to take a mixed methods approach because music is so multifaceted. And for quantitative data, I used two APIs. So I was measuring quantitative audio features from Spotify. Spotify for each song has a set of numbers that measure like energy from 0 to 1 or the tempo of a song. An API, which was what I was using, basically is a go-between to get data from a website like Spotify into my program that I was using for analysis, which was RStudio. I also wanted the song lyrics for quantitative analysis, or qualitative. So I used an API to get the song lyrics from Genius as well. Special thanks to Cameron Mulder at UO Libraries Data Science, because he was a big help with figuring out using these APIs. I have some preliminary findings for both sets of data. So for the quantitative data, just some basic things like explicitness, modality. So country here on the left is my control group. So you can see that pop is much more likely to be explicit than K-pop or country, and country is much less likely to be major, or it's more likely to be major. There were almost no minor country songs, while the other two genres were more similar. So going into this, my hypothesis was that there wouldn't be a significant difference between pop and K-pop because, they're both pop related. And my theory was that these lines are drawn because of race-related reasons. But my data said otherwise, which was really cool to find out. So for these three specific audio features, so danceability, which is like engaging with the tempo and like the dynamics to measure how danceable a song is. Speechiness measures how close the song is to a spoken word track. So like a rap song would be like a 0.33 on the scale. And then energy measures like the general energy of the song. And so for all of these, K-pop was higher, the mean value was higher for K-pop using the song samples. And this raises a lot of other questions, like why is it higher in all of these things? And I don't have time to exactly go into why, but that's something that I'm looking forward to investigating more into. I had a few surprises. So valence, which is generally how happy the song is, and acousticness. The relationship between country and K-pop, when I ran some statistical tests, was not significant. And the relationship between country and pop for acousticness was not significant either. So remembering my hypothesis, it's really K-pop and pop that I would expect to not show significant differences. So to find these other relationships raises a lot of questions about what in this music is making it like this. But anyway, moving on to the qualitative side, I used a blend of thematic and content analysis. So I developed like a multi-step coding process. I used NVivo, which is like a qualitative data software, and you can add labels and tags to different parts of the lyrics. So I did that for 30 songs from each genre and just found some preliminary things. Country, it was big on, I had to add a category called rural life because there's so many references to like tractors or farming. So that was very unique to country. They also talked a lot about the past and had half of the songs referenced alcohol, usually whiskey or beer. So that was like my control sample, but we get a really clear picture of the country music genre. With pop and K-pop, they were more similar. You can see there's love relationship was significant in both of the samples. Breakups was a big part of the pop genre. Also a significant amount with alcohol and drug references. But something that was really interesting to me about K-pop was 19 of the 30 songs reference confidence of some kind. So like either addressing haters or like encouraging others to be confident in themselves. And sometimes this went further. So I had to create this label called like swagger, which just encompasses like bragging and toxic confidence, which these two were barely present in the other two genres. So it kind of raises some questions about why are K-pop artists singing about confidence and what does that have to do with K-pop's like global popularity? So basically looking ahead, I want to run more. So I've just been doing exploratory data analysis, but to describe what happened in the data, I'm looking to move from that to more real data analysis in R for the quantitative side. And then for the qualitative side, I want to look back more closely at some of the song excerpts and do selective coding, which is part of grounded theory. So yeah, thank you for listening today. I hope this was somewhat informative.
Thank you, Amelia. Okay, moving forward, we have Mary, who has recently graduated. So congratulations and thanks for joining us.
Thanks. All right, can you see the slides okay?
Yes, thank you.
Okay. All right, like Lance said, my name is Mary. The project that I worked on this summer was called oocyte meiotic division, cortical stiffness versus cortical contractility. I completed this project in the Bowerman lab along with my mentor, Alyssa. So before I jump into the project, I want to give a little bit of background information. First off, meiosis is the process of cell division that creates sex cells. Female meiosis in particular involves two asymmetrical divisions. So unlike in mitosis, where the chromosomes and the spindle are in the center of the cell, in meiosis, they actually migrate to the cell cortex, where half of the chromosomes are extruded into a polar body in meiosis 1, and half of the sister chromatids are extruded into a second polar body during meiosis 2. This leaves the resulting ovum with most of the cell's resources so it can develop further on. Any errors in this process can lead to developmental defects, so our research can help with understanding these errors. The part that we were most interested in was meiosis 1. So during meiosis 1, the chromosomes, which are in magenta here, migrate over to the plasma membrane, shown in cyan. Here, the contractile ring begins to form right by the spindle, and active myosin patches begin to appear on the cortex. It is thought that these patches provide the contractile force that pulls the contractile ring inwards past one set of chromosomes to extrude a polar body. And while this happens, we also notice smaller furrows forming throughout the plasma membrane. These smaller furrows are what we focus on in the project. So how can we look at this furrowing in vivo? We use C. elegans oocytes, and C. elegans are a great model organism because they are transparent, which you can see right here. This allows us to fluorescently tag proteins via CRISPR to visualize different structures, such as the plasma membrane, the chromosomes, and microtubules. So once we had our worms ready, we could then live image their oocytes using a spinning disc confocal microscope. This allowed us to get movies such as this one. Here is a control oocyte. Again, the chromosomes are in magenta and the plasma membrane is in cyan. And as you can see, the chromosomes migrate over to the cortex, furrowing begins, and one set of chromosomes is captured and extruded. There's a mutant that lacks the protein CLS2, which stabilizes microtubules, and these mutants show an increase in furrowing. And I don't mean the main furrow that captures the chromosomes, but these smaller furrows. We quantified this data in two different ways. First, by taking the mean length of the furrows in microns over time in seconds after anaphase onset. Anaphase is when we observe furrowing occurring. So the control is shown in white here, and the CLS2 mutant is shown in purple. As you can see, there is a clear increase in furrow length when looking at the mean length. Secondly, we looked at the sum of furrow lengths, again in microns, over time in seconds after anaphase onset. And similarly, we see an increase in furrow length. So why does this happen? Why do CLS2 mutants exhibit excessive furrowing? Well, we already knew that CLS2 had TOG-like domains that stabilize microtubules. And before the lab even looked at the plasma membrane, they first looked at microtubules, which are shown in green here, as well as chromosomes. And while looking at these movies, you'll notice that in the control, there are these brighter patches that show up at the beginning that the mutant lacks. So with those two pieces of information, we hypothesized that cortical CLS2 patches stabilize cortical microtubules to provide the stiffness that counteracts membrane ingression caused by actomyosin contractility. So to investigate this hypothesis, my job was to answer this question. Do increased cortical microtubule levels suppress excessive membrane ingression in CLS2 mutants? And to do this, I used RNAi to knock down either CLIP7 or ZIG9, which are both microtubule regulators in CLS2 mutants. The goal here was to restore those microtubule levels, and if that does indeed suppress furrowing or excessive furrowing, that would support our hypothesis. So once those strains were ready, we could then live image the oocytes expressing plasma membrane and chromosome markers. And then lastly, we collaborated with Adam Fries to quantify the furrows. So we basically detected any defects in the hull, or in our case, any furrows in the plasma membrane, and then we were able to measure the furrows any way we wanted to. So in the end, we found out that knocking down either CLIP7 or ZIG9 does indeed reduce excessive furrowing in these mutants. So on the left, I'm showing the CLS2 mutant data in purple versus the CLS2 CLIP7 double mutant in pink. Again, looking at the mean length of the furrows and the sum of furrow lengths. In both of these, you can see that there is a reduction in furrowing, which supports our hypothesis. Increasing microtubules reduce that excessive furrowing in the mutants. Same thing is seen when we look at the mean length in CLS2 Zyg9 double mutants, which is shown in blue here. However, there was not a significant difference when looking at the sum of furrow lengths. However, this may be because the double mutant has more smaller furrows, which would reduce the mean length, but take that sum of lengths way up. So with this data, we were able to conclude that increasing cortical microtubules rescues furrowing, which again supports our hypothesis. So with that part of the project wrapped up, we moved into exploring the mechanism, trying to answer questions such as where are microtubules and myosin relative to the furrows. We also investigated other regulators that were upstream of CLS2 that may be involved in regulating membrane ingression. And here's an example movie of BUB1, which is upstream of CLS2. You can see that it also forms patches on the cortex. But like Lance said, I've graduated, so this next part of the project will be completed by whoever picks it up next. And lastly, I want to thank Cure and both the Bowerman Lab for supporting my research this summer.
Thank you, Mary. Okay, next up is Siri.
Hello. All right, so my research project is on the role of language planning in speech and breathing coordination. And I did this in the University of Oregon Speech and Language Lab under Dr. Melissa Redford. So I have been researching how the process of speaking is inseparable from the process of breathing, as air must flow through your larynx and your oral and nasal cavities in order for you to produce speech sounds. So these two processes have to be coordinated because you're not able to produce speech while you're inhaling. And so you have to time your intake of air with your thoughts and match the breath to your length and intensity of your speech. You have to alternate between these two phases, which you can see on this little picture I drew on the right under speech breathing. So you start with an inhale, and then you have time to speak, and then you have to pause while you're inhaling before your next round of speech. So these pauses are considered grammatical in fluent speech when they occur after a completed thought. And there's evidence to suggest that adults prepare their inhalations so that the exhalation phase matches the length of their utterance or their completed thought. So you're basically planning your speech with a breathing framework as you go. And this is what my research is looking at, the connection between speech and planning it, as well as breathing. And when the language planning process is interrupted, whether breathing also becomes less grammatical or well-timed because it's a part of the language planning process. So I'm using respiratory inductance plasmography, or RIP. This involves using the machine here on the left. And I fit the participant with two bands, one around the chest and one around the abdomen. As you can see, I'm showing it on me in the right picture. And then these bands are elastic and they can detect respiratory movements, which affects their inductance. The program I'm using converts these signals into a readable graph, and the amplitude reflects the changes in your breathing and the changes in your lung volume. So this is also aligned with the speech that I have the participant do, which is recorded on an audio graph. And so I can look at where the breaths fall, as well as how big they are in comparison with the speech. So for my actual stimuli, I'm using 5-minute animations from Pingu, which is a claymation series. I have the participant sit down and they're all hooked up into the RIP system and they watch the video. And then I have them retell the story as images from the video are shown to them. So they're given a visual aid like this clip in the middle here. And then while they're telling the story, they have to remember a sequence of colors like this on the left. And so this is what is disrupting their language planning, because it is giving them an additional cognitive task to do while they are recalling the story. And then they are interrupted from telling the story and asked, what was the color sequence? And so this tax on the language planning process is what I'm looking at, and I'm seeing whether it has an effect on the speech and breathing coordination. I'm also running an expository speech task where participants have to explain how to do a series of tasks around the University of Oregon as if they were talking to a new student. So they have to do this, and this is an example of what they might have to explain. So they're given different tasks around campus, and then they're basically just giving me an explanation of how they would do this. And I'm also having them do this at a very fast speaking pace, so that I'm looking at whether this is an additional cognitive load on speaking and whether their breaths change in this as well. So for data analysis, I am manually going through the data, and I'm transcribing the speech, which is shown in the bottom graph here. And then this is a graph of the breathing. So the green is inhalations and the purple is exhalations, and they line up with the audio, as you can see. I'm still in the middle of gathering data, but I'm also analyzing as I go. So far I've noticed that inhalations fall grammatically during natural and untaxed speech. And this is as we would expect. And they become more urgent and less grammatical when they are taxed with an extra cognitive task. So I have an example here of an ungrammatical breath, which is what I'm looking for. So the participant starts, can you guys see my cursor? The participant starts with an inhale, and then they're giving me a sample of speech here. During the exhale, they say, you're gonna cross another street you're gonna cross another intersection. Look both ways before crossing, and you're gonna walk past it, walk past the University of Oregon Law School, walk past, and then they have to take a big inhale because they've run out of breath. And then they say and then they do another little breath, and then they continue their sentence. So this interrupts their thought, and so that's an ungrammatical breath. So I'm looking for these all throughout the audio clips, which is about 30 minutes of speech that I'm picking through. So this is so far indicating to me that the language planning process does involve a breathing framework planning subconsciously. And so as the language planning process is taxed, inhalations become more need-based and not as well planned, which is showing me that breathing is woven into higher level language planning. And I'm really excited to see if this is consistent. And as I go forward, I will keep bringing in new participants and having them run these two tasks. And I'm looking forward to doing this as fall starts.
Thank you so much, Siri. We are going back to learn a little bit more about, we're going back to music now. So I give you the floor, Sue Carney.
I'm not sure how to access sharing my screen. Oh, here we go. Sorry, guys. Okay. There we go. All right. Okay. Can everybody hear me? Okay, great.
Yes, we can see.
Okay, thank you very much. Good afternoon, everybody. My my project has to do with issues in delivering music therapy to patients in hospitals. There's some question as to whether or not it is necessary that the therapist be present for the therapy, or if there's some sort of emotion that that is conveyed, or some sort of connection that is made by having live therapist and live music, versus being able to deliver via telemedicine, which there hasn't been a lot of experimenting with that, and not a lot is known about it. But of course, we had a wonderful opportunity when we had the laboratory that was created by COVID-19. So briefly, to describe the situation, the use of music in hospitals has been going on for a long time. As time has gone by, around the turn of the century, more and more tests and studies have been done on the situation with using music in hospitals. It can be used to alleviate pain. It can be used to treat anxiety. It can help with regulating heart rate. And it's especially useful in NICU situations. It can really help to bridge. So all of that information about how music has been useful in hospitals, there's a ton of information about that. What we really wanted to look at, though, with this was to see what would happen if you couldn't go into the hospital, and whether or not the treatment would be as the efficacy would be the same. Okay, so the research question basically for me morphed into, are there differences in the efficacy of live music therapy versus synchronous remote music, which means remote music that's happening simultaneously, as opposed to asynchronous, which is recorded music. So are there differences in the efficacy of this? And what I was talking about a little bit earlier is we're looking at therapeutic presence or psychological transference, depending on whether we're looking at this from the standpoint of music therapists or of other music professionals. And we want to know, can remote treatment be prescribed with confidence? Because we just had this pandemic, and what would happen if there was another pandemic? And also, there's a lot involved with putting a musician into a therapeutic hospital situation. They have to bring musical instruments, they have to travel from room to room, they have to travel to the site, and so on and so forth. Is it possible that remote treatment would be a viable option in some cases? So this piece of work that I've done here is called a scoping review. Many of you may be already familiar with a scoping review, but it's a relatively new form of review. It's a form of systematic review. Now, a systematic review and to a greater extent, a scoping review has to do with more general questions that, unlike the very specific research questions that have been asked here that we've seen the presentations for, this involves a lot of very beginning background searches. And as we delved into the literature, we found that there was not a lot of literature available. There has not been a lot of experimentation. And very quickly, as we chained through with our data, we found that there were only a few articles written specifically on this subject. In fact, we were able to come up with about 48 articles total. Now, we used different search strategies for different sources. And in a scoping review, you can use more different sources. A lot of especially for this, a lot of the work that's being done on this is so new that we were able to go into things like thesis statements and stuff like that, so that we were able to search through books, all sorts of things other than the regular articles, the peer-reviewed articles. You can see here an example of our search strategy. This was for PubMed. PubMed is a database of medical articles, and they have different search terms than what we would usually use. But these were constructed for me with the help of the reference librarian. Now, we went ahead after we found our initial references. We went through the literature, as I said, using reference and citation chaining, and we uploaded all of this to Zotero, and now we're using the Covidence platform. one of the things that made us choose a scoping review is because a scoping review can be done with a single reviewer. If we were doing a full systematic review, it requires more than one reviewer. And in this case, we did not have that. Now, here are some of our references. Because of the nature of this kind of a study, we went through a lot of references. I think there's 34 shown here. I'll take a look at, we can take a look at some a little bit closer here. These are representative. Now you'll notice that here we find another scoping review. These were very useful to us, other reviews. Meta-analysis is an example of an article that would be good for us. Feasibility pilot study, you see, this is not necessarily a published article. And this understanding the technological practices and needs of music therapists, this is based on a survey with music therapists. And so it's just insights into what they might need. Now, we are right at the point where we're getting ready to analyze the data. This presentation came a little bit early, but our early results, and in keeping with the usual goals of a scoping review, we did find that there is need for further study on the question of whether efficacy with remote telemusic therapy. Right now, there have been very few studies, as I said. The question, though, is highly relevant because a lot of medical treatment depends on whether or not this can be done remotely. It adds flexibility, as I said, to the practitioners. And it also has commercial implications because perhaps the music can be more mainstreamed into medical practice if it is not such an involved and expensive thing to get into the hospitals. So the stakeholders that we've identified are, of course, the patients, music practitioners, hospital and medical staff, and we could all just use to have music more mainstreamed into hospital treatment. That's a personal goal of mine, is to try to do some research that will lead to the greater inclusion of the music that we have learned is so useful. I've also identified some other lines of inquiry. We also want to look later at recorded music versus synchronous music. Also, curiously, we ran into quite a bit of writing about the use of artificial intelligence and robotics, which might seem completely antithetical to the idea of the therapeutic presence. But we want to see if perhaps robotics can prove useful in this. We are also wanting to look at the physical qualities of sound. By that, I mean, not just the fact that there is music and music has its musical qualities, but also if music is coming through a speaker, what does the quality of that sound have to do with the quality of the therapy? There's really been only a little bit also researched on that. And finally, what role does environmental sound play in the therapeutic delivery and effectiveness? What got me into this study, this particular study actually was I spent time working as a therapeutic musician in hospitals. And a lot of these thoughts occurred to me while I was doing the work, in particular, the one about environmental sound, because if you've spent any time at all in a hospital, you'll recognize that the environmental sound there is pretty out of control. That is really all that I have to say at this point. I'm looking forward to reporting more once we have gathered and extracted the data, and hopefully we'll have something meaningful to say. But in the meantime, I'd like to thank to everybody who contributed to this project. This is a wonderful opportunity, a wonderful chance to do some research. Perhaps not such a wonderful chance to speak publicly, but I thank you all for listening, and I turn it back over.
Thank you so much, Sue. And, through all of these presentations, I hope that you stay connected with one another, because I see some related topics where you can really continue to rely on each other's energies and insights and connect, and maybe kind of form your own academic research club, right? The Cure Club, where you can all continue to stay connected with one another. Okay, moving forward, we have Lily. Lily, are you ready to share your screen?
Yep, great.
Thank you.
All right, give me one moment here. All right. So my project, I actually had two projects I worked on over this summer. My first project was improving DESI-MS imaging of tissue samples. And my second project, which I kind of picked up around halfway through the summer, was improving mass spec file processing. So first, let's get into DESI-MS imaging and why you should care about it and how it's important and relevant. So DESI-MS imaging is a new way of imaging tissue samples, which allows us to see proteins in these tissue samples. As you can see right here at the bottom, I have an optical image of a rat brain tissue slice, and I've used the DESI-MS imaging processes that I've developed over this summer to image a protein myelin basic in the sample. And you can see exactly where the myelin basic is in this tissue sample. This is applications in histology, in research, and medical fields. I'll get into those applications a little bit later. But this is a very important new field of histology that I'm helping develop that will really be useful for a lot of people. So let's go over what DESI-MS imaging is. So DESI-MS imaging is a technique that's been developed by the Laskin group. And how DESI-MS imaging works is you take a high voltage source and you ionize small pieces of a tissue sample. And each one of those small pieces of a tissue sample goes into a mass spectrometer, which then we can analyze using our my group, the pro group's in-house software IFAMs. So a mass spectrometer, how it works is you intake these ions, and what the data that you get out from it is your mass over your charge state and the abundance at each of those mass over charge states. So what this is basically is it gives us the abundance of masses at different points. So we're able to see like the mass of a protein and how much there is, or an element, or things like that. So once we get this mass spectrometer data, we use IFAMs, and in IFAMs, we use something called Gabor transformation. And what Gabor transformation is, it's a different way of looking at this mass spectrometer data. So I'm not going to go into Gabor spectrograms and Gabor transformations that much, as it's a lot. But what you need to know is, you can see I have these boxes selected here, and these boxes show the spacing between peaks, and they show these natural curve lines. And what we can get from this by selecting out these natural curve lines and these spacing between each of these different peaks is all of the charge states of one protein. So we're able to basically get all of the M over Z's of one protein, and we're able to, using deconvolution, spit out our final abundance at one mass. So this basically, all of that to say, basically allows us to get where, like, how big our proteins are and how much we have of each protein in our sample. So that's a lot of background and not really what I've been working on over the summer. So what I've been working on over the summer is actually speeding up and improving how these images are made and created. So initially, for our lab to create just one of these images that you've seen of these tissue samples, it would take 2 weeks of all my lab working hands on deck to get these images to be made. And what I did over the course of the summer was develop a pipeline and use multi-processing in conjunction with UO's Tilapis supercomputing cluster to get these images created down from 2 weeks to like 5 minutes, which is just a crazy jump, and is great because it allows us to create these images really fast, easily, and readily. So you don't have to worry about accidentally imaging the wrong protein, and then you have to spend another like 2 weeks to make one image. This is the group that we're collaborating with, the Laskin group, plans to use this in medical applications like time-sensitive surgeries, especially in removing cancers. They specifically plan to use it in brain cancer, brain tumor removal surgeries, where you have to make sure you get rid of all the cancer and you have to do it as fast as possible. And this will allow them to image the tissue samples and see exactly where the proteins of that cancer are located and remove that cancer efficiently and quickly. This could also have applications to your life in the future. You could, let's say, have a mole that looks, you might think it's cancer. So you go into the dermatologist, and the dermatologist takes a tissue graft of that, puts in the mass spectrometer, and before you even leave, you could know if that tissue sample was cancerous or not. This is a better approach than than staining, because it allows you to individually image each protein, and you don't have to worry about how long staining takes or that you're staining multiple proteins or anything. You can very specifically pinpoint very accurate proteins. The Laskin group had a rudimentary way of imaging, as you can see on the left here. These are two images of the same protein, myelin basic. And as you can see beforehand, they had this very noisy image that you couldn't really get a lot from. But then after the improvements that I've made to the software, and also my grad student, Cade, he implemented something called background subtraction into this code, we're able to get these much clearer, higher resolution images that show us exactly where proteins are located in a tissue sample. This also has applications in research and all types of field of research, anything that's looking at proteins, any medical research. This is going to be huge for a lot of groups because they're going to be able to look at where proteins are located in tissue samples much easier. Yeah, so my work has been really, really important in developing this new method of histology, and beforehand it was very impractical to create these images at all. So moving forward, also, my other project that I got caught up in over the summer was processing .d ion mobility files. So ion mobility is this new, I mean, it's not new, but a lot of machines nowadays are coming out with the ability to have ion mobility data in your mass spectrometer data. And ion mobility is basically another dimension of data that tells you how far ions moved, which can be used to separate overlapping peaks from each other. But the current software that's available to read these ion mobility files that the mass spectrometer makes aren't very good. They don't have capability to batch process or process multiple files. They don't have the capability to do a lot of things that a lot of research groups are trying to do, and everything has to be done manually, which is very slow. So my project was developing a piece of software that's able to do all these things automated and able to do a lot of new features that the current software of reading these files isn't able to do. This is a collaboration with CHOP, which is Children's Research Hospital of Philadelphia. And CHOP is planning to use this data to develop new methods of detecting diseases so that they can, well, find these diseases easier and detect these diseases before they become symptomatic. This would be something that your blood could get tested for instance. These images that I have here down at the bottom are mainly just to show that I've replicated software that is currently available to us. And that is about it. I know that's a lot of intro in 10 minutes. So if anybody has any questions, I'd be happy to answer them.
Any questions for Lily before we move to Olivia?
Well, great. Thank you so much. I'd like to give a thank you to the Pro Group and everyone there. And I'd also like to give a thank you to Agilent. They were a company that I collaborated with and gave me a lot of help with things. Thank you.
Thank you, Lily. Okay, up next is Olivia. Drugs influence the musical landscape of the 60s. All right, the floor is yours.
All right, just gotta pull up my screen. I always forget how to do it, even though I literally did it earlier today. Show all windows. Okay, can everyone see that? Yeah, can everyone see my presentation?
Yes.
Awesome. So my original title, How Drugs Influenced the Music of the 1960s. It actually included the early 1970s as well, but I've really been wanting to find where I want to focus. So I decided to cut that out a little bit, at least for most of it. I'm probably going to touch on it a bit, but my title is going to change because the direction of my research has changed in light of new evidence. And also I just want to give a disclaimer that my presentation's pretty loose because it's mostly it's history, and history is quite messy. Maybe not as explicit as I would like, but still exciting stuff. So anyways, first I want to talk about how I got here. So I did the first half of this research, or a little less than half of this research, in winter 2021 for my required history 407 seminar, where I mainly focused on a question of whether drugs had a causal relationship with changing music trends. In that original paper, I described, I suppose, chronology. So I started with the kind of a basic history of drugs in the US, and by extension, the United Kingdom, because in the 1960s, of course, you have the British invasion and everything. And in that, I touched on the development of birth control for really kind of expanding the rights of women and their ability to participate in more as time went on, as well as the distinction that I was noticing about the words dope and drugs in reference to drugs. So I've explored that a bit. So then I start talking about the early years, the early 1960s, and by extension, the late 1950s, because they were really similar musically. Very sanitized and I guess kind of a lot of music back then was kind of fitting a mold, but then you start to see a change in the mid 1960s, especially with the Beatles are kind of like my blueprint a little bit when it comes to how pop music changed, and it was very evident that many pop phenomena followed their footsteps. So we see in the late 1960s, different music festivals focused on psychedelia, free love, and then I noticed that it kind of winds down a little bit towards the end of the 1960s, coincidentally, as there are a few really prominent deaths in the music world. Some of them related to drugs, some not, but I did notice a lot of correlations between how drugs were referenced in music, used in mostly the counterculture, and how music innovation changed and music trends in general. So I've been building off that work and expanding into larger, maybe more socially aware questions, because the 1960s it's a really big part. It's a lot to unpack. So, and that goes into the reasons why I chose this topic in general. The 1960s is a really unique time period to look at in a lot of ways. I knew I wanted to ask something that tied the music to an important social issue of the time, or I guess social issues, because there were a lot. Examining the context surrounding music tells our stories throughout the years, and in turn, stories of change, which I find fascinating and really eye-opening into understanding the past. So my methodology includes a lot of different avenues, which makes it even more for one person, but a lot of my primary source material has been lyrical content and the music itself, because as a music historian, that's really the first place I'm going to go. And so far, I've tried to use also a variety of secondary sources from different fields, including the sciences. So mostly sociology and psychology, because I feel like those fields give a lot of insight into why things happen over time, why people act a certain way over time, things like that, and drug history, of course. Music history, political history. So I've tried not to focus too much on one area because the topic is so complex. over the summer, I focused a lot more on journalism and media, particularly interviews, articles, even photos and video. Interviews especially have given me a lot of insight on, on perceptions of certain groups. For example, the San Francisco hippies versus the equivalent demographic in Los Angeles. I learned through watching a film about the Monterey Pop Festival in 1967 that there was actually kind of a feud going on between those two groups, and I was under the impression that the country was pretty much united in that way in terms of the counterculture, but even within the counterculture, there were subgroups that were at odds with each other. And as an extension of that, one thing that I really want to work on going forward is seeing if I can find evidence of that in any of the music at the time. And I also, as I mentioned in a previous meeting, I met with an individual I know who actually attended the Woodstock Festival in August 1969. I did one interview with him, and we intend to chat together again soon because they realized that they have a lot more to say, and there is a lot to say about experiencing such a impactful social phenomenon. And I've spent quite a bit of time this summer learning about how to conduct interviews because I've also been doing an internship through the Oregon Folklife Network, how to conduct ethical fieldwork with traditional artists and by extension, people who have just basically people who have stories to tell. So I've gotten a lot better about, I guess, more experience with conducting interviews in that way. And then my last point is I've also taken a lot of looks at individual musicians and their relationships with drugs, as well as how their music has changed at certain points in time. I've noticed the most change during critical points, such as about 1965, 66, and then 67, and then 69. So in my research, those have those turned into hotspots, and they happen to align with certain historical events, at least in the latter two, with the two, the Monterey Pop Festival and the Woodstock Festival. So nitty gritty, or I guess to the fun part. So I actually did a trip this summer with some of my funding. I traveled to Cleveland, Ohio, and I spent 2 days in the Rock Hall archives, which so how it works is they accompany the Rock and Roll Hall of Fame itself, and they're part of the museum. They're not in the same complex or anything like that, but they're actually part of a community college campus. But the Rock Hall archives preserve and digitize things that will be displayed in the museum. So I got a chance not only to visit the museum and see what they had on display there, and it just so happened they have a massive exhibit on the Beatles right now because they just the remaining Beatles released a series of sessions called the Get Back sessions, I believe it is. But anyways, yeah, I spent 2 days right here looking at, my favorite was the periodicals. So I looked at a lot of different, mostly independent journalism, but I also looked at different publications that specialized in popular music like Motown, rock and roll, folk music, those kinds of things. And one unique thing I didn't realize was that I had to be very careful with the material because it is 60 years old. Yeah, 60-year-old material. But it was a lot of fun, and I got a lot of material to sift through, ranging from very polarizing political cartoons, you know, bashing certain groups, members of the counterculture, or like, you know, other kind of parts of the political spectrum. But I also found poetry, interviews, opinion pieces, a lot of different things. I also watched a lot of archival footage, outtakes from Woodstock, a whole movie from the Woodstock Festival, a whole binder full of pictures of Eric Clapton. Just yeah, they had so much to offer, and I'm still working through all of that. So and then lastly, I also read through research notes from Rolling Stone. So the clippings and photographs and articles and things that they would be using to support an article that they were writing at the time. And some of it was very helpful. So, and yeah, I already talked about all this, but these are a few of my images that I was able to take. So on the left here, I see it was really hard to read, but it's basically like a satire piece about laws continually being made against dope or you know, fun drugs. And then on the top right here, that was from a more independent publication, kind of talking about the kind of return to the earth movement. And then the bottom right here was a very interesting interview between Rock and Roll Magazine and Berry Gordy, the founder of Motown. So this article in particular actually made me start thinking about the relationship between musicians and journalists, and by extension, media as a whole. So that's it. So that brings me to my current progress. I already mentioned that I am sorting through a lot of material. And because this is my honors thesis, so it's a very extended project, not just including the summer. I mostly use the summer for digging and searching and finding stuff, and now I'm using this time to outline all of my information. And let's see. Yeah, so I plan to take my previous paper on me investigating that causality or that correlation between music and drugs / dope, and just turning that into a chapter as a smaller story, and then adapting it into my bigger story of and I think my bigger question has to do with authenticity, building off this tension between maybe the more radical groups and musicians, and then maybe there are more oppositional journalists and rock critics that I found quite a bit of evidence for, and turning that into a argument about authenticity on both sides of that spectrum and what it means be authentic, and how music tells a story of authenticity in the 1960s, and probably we'll kind of bring that to the present day, just so I can have that bit of relatability. But yeah, my research question is changing right now, so I'm at a very interesting stage right now. But in the end, I feel like what it comes down to is the music and how it reflects change, and I'm really excited to continue working on this and eventually defend my thesis and present for the symposium. So that's all I have, but if anyone has any questions, I'm happy to answer.
Thank you so much, Olivia. And that's what research is really all about, right? Asking questions and going with the ebb and flow and being flexible depending on the path of your research. And I think that you've all gotten a snippet of that this summer that you will be able to continue with. Okay, Leah Blankenship, you are up next.
Great, can you all see my screen okay? Awesome. Great, so my project was on the anatomical and functional differences between CCK positive cells and other TAC1 positive cells. I did this project in the Sewestrick lab in the Institute of Neuroscience. And Dr. Sewestrick has been mentoring me, and it's just been a fantastic experience. So the Sewestrick lab focuses on the habenula, so this is specifically talking about the habenula. The medial habenula is a really small part of the brain. It's part of the epithalamus, so it's kind of in the middle of your brain, right near the ventricles, which is where the spinal fluid moves in the center of the brain. The medial habenula is involved in a really wide variety of behaviors. Kind of most of them are listed here that different papers have found so far. The medial habenula is thought to potentially result in decreased dopamine receptor and transporter expression due to that fact and a bunch of the behaviors listed here. It's also thought that it could play a role in behaviors and conditions such as schizophrenia and attention deficit hyperactivity disorder. So this work has a lot of implications for human behavior in general, and then a lot of specific conditions as well. So we're interested to know what cell types in the habenula are involved in what of those long list of behaviors. It's a very heterogeneous cell type population in this area. So previous work in the Sewestrick lab has looked at these TAC1 cells that are part of the medial habenula here. And previous in situ studies showed that those TAC1 cells are mostly located in the dorsal medial habenula, and the CCK cells are as well, which is what my project is focused on. Ablation studies of the dorsal medial habenula, where that part of the brain was ablated in mice and rats, suggests that that part of the brain is probably involved in reward behaviors, exercise motivation, and regulation of hedonic state. And within that, those ablation studies, as you can see in these images here, those TAC1 cells were an important population of cells that were reduced. More recent single RNA sequencing studies identify CCK as a potential putative marker of the dorsal medial habenula, as you can see in this diagram on the right. That population is very much present in that dorsal medial habenula population and not really some of the other parts of the medial habenula. And the CCK cells are then thought to potentially be a subset of TAC1 cells, which the Sewestrick lab has looked at in depth. So that takes us to my project. The questions I've been looking at are, how are CCK positive cells different from other TAC1 positive cells? That means I'm going to look at, are they for sure a subset of the TAC1 cells? My second question is, how are they anatomically different from the TAC1 cells? So are they a randomly arranged subset, or is it more specific? And then do the axons all go to the same place, or do they project somewhere specific, different from the rest of the population? And then how are they functionally distinct from the TAC1 cells? So we know from other research in the Sewestrick lab that TAC1 cells are active during reward-guided behavior, specifically when reward is expected and then withheld. And CCK positive cells are active. We want to know if they're active during those same times or involved in other behaviors. So to look at that first question, I did some in situ hybridization studies, which is where we look at the RNA expression overlap between CCK and TAC1 genes. So these are some images from my first attempt at this, which was a few months ago. These did not turn out very well. As you can see, the green stain that we used, the antibodies, the fluorescence for the TAC1, ended up kind of lining up with a really heavy green autofluorescence from the tissue. So it's kind of hard to tell what is actually TAC1 cells versus just general autofluorescence from the tissue. So I'm going to be trying this experiment again in the next 2 weeks to kind of take a look at this question further and hopefully get better results. For our second question, looking at the anatomical differences, we're tracing the axon projections of these cells by clearing the tissue with a clarity protocol, which is where we basically remove all of we perfuse the tissue with like a monomer solution, remove all of the fat from the tissue and polymerize that so the tissue holds its shape, but all of that opaque like myelin and stuff in the tissue is kind of removed so we can image those axons for really large tissue volumes. In the middle are these TAC1 images that Emily took as part of some previous work. And then on the right are my CCK images that I actually took last summer. And as you can see, the TAC1 axons come down and kind of arborize in the interpeduncular nucleus, the IPN, whereas it's really easy to see in that like sagittal image on the side there, but you can see that those CCK neurons come down and kind of just move right over the IPN there, which is really interesting. So it looks like the axons do go to a different place. I attempted this experiment again this summer to try to get some images further back in the brain and see where those axons go. We tried to take a look at those images this week, and the experiment did not turn out exactly how we hoped that it would. So unfortunately, I'm going to have to try those experiments again, but that's the process of science. So I was able this summer, though, to get cleared on surgery training. So for these next round of experiments, I will be able to do all of the viral injections and surgeries for these mice myself, which is really exciting. But yeah, I will be doing those experiments again. For the last question, which is about the behavior aspect of my experiment, we're looking at those functional differences. For this, we did fiber photometry during a standard 3-choice reaction time task. So here, these animals undergo a viral injection into the frenula that allows for GCAMP expression in CCK positive cells. And then that GCAMP fluoresces when calcium is present in the cells. And then when we implant a optic fiber above that injection location, we're able to monitor that fluorescence as the animal is going through these behavior activities and use that fluorescence as an indicator of the activity of those cells. So the animal has to make a choice between these 3 ports on one side of the behavior arena, and when a light turns on, and if the animal selects the correct port with the light, then the animal gets a reward of sucrose water on the other side of the cage. That's how the behavior worked. And then we were recording, like I said, that fluorescence activity to keep an eye on what the neural activity of those CCK positive cells looked like as the animal went through these behavior experiments. I trained these animals all through June and July and the beginning of August, and was finally able to collect some data from them the past few weeks. So here you can see these plots. We're interested in knowing if they respond to withheld reward, similar to the TAC1 cells, like I said before. So occasionally when the animal would do all of the correct behaviors, poke the light at the right time, go to the reward port at the right time, we would occasionally not administer the reward. And then we would look at the neural activity when that happened. And these kind of purple colored plots here, each little line through the plot represents a trial, and the color represents the fluorescence during that trial, with a more yellow color representing a Z-score of activity further from the baseline. So you can see in these 2 withheld plots, for the first one is for a reward probability of 50%, and then the second plot is for a reward probability of 80%. You can see in both of them right after that 0 time mark when the reward is expected but withheld, there's like kind of a line of really faint line you can see of that increased neural activity, just some more of that yellow color. And then on those bottom graphs, you can see that black line represents when the reward is withheld, and you can see there's like increased activity over the rest of the trials where the reward was delivered. So it does look like these CCK cells are active during withheld reward, similar to the other TAC1 cells. It also looks like there might be some difference in that activity depending on reward probability, which would be something interesting to look at in the future. We're also interested in knowing about how CCK cells respond to aversive stimulus. This figure is from another paper that administered like a foot shock to animals and saw an increase in CCK cell activity during that. And so we were interested in wondering if CCK cells respond to aversive stimulus because TAC1 cells do not. So for our experiment, we administered an air puff. Sometimes when the animal would go to the port, it would get an air puff in its face, which they really don't like. And we would also monitor the neural activity while that happened. So you can see in this plot on, you can see that the air puff, we didn't really see a strong response to the air puff as compared to the like the withheld reward. You can see in the top right here, there's a figure that kind of summarizes both of those figures from the previous slide. You can see that really noticeable line of all those yellow trials that represent more neural activity in response to that withheld reward, but we don't see that on the air puff plot. So that kind of looked like there probably isn't a response to aversive stimulus, which would suggest that the CCK cells have a function pretty similar to the rest of the TAC1 cells. This did contradict this other paper that suggested that there was activity, but there's a lot of reasons for why this could happen. The other study used extremely different methods to look at the cellular activity. Like I said, they used a foot shock, which is obviously a much more aversive stimulus than an air puff. And also they were looking at FOS expression in the tissue ex vivo, so not while the mouse was alive. They're just looking at the tissue samples afterward and that FOS expression as a proxy for activity. So very different methods to look at these different things. However, we did see, so these are all plots from individual animals in my experiment. So you can see both M1612 and 1613 had pretty similar, no response to air puff, really limited response to withheld, but still enough to kind of show up on those graphs. But interestingly, in M1610, we did see this really distinct response to the air puff, which we didn't see in any of the other animals, both in this behavior cohort and in previous behavior cohorts that I have run. So that was really interesting. We also didn't see, you know, super strong withheld reward response, but did see some. But we're really interested in the fact that we saw an air puff response in that animal. So this next week, I'm going to be doing histology on these tissues to kind of see if those injections and the implant were located in the correct place to see, you know, if this data really is representative of CCK cells in the habenula. There is another region of the brain that's a little more ventral of the habenula that does have a CCK positive cell population, and that does respond to aversive stimulus. So it's possible that this injection and implant was like slightly too ventral or something like that. But I'm going to take a look at that later this week and hopefully figure out how to interpret this really strange result in this one animal. But that was in general, between, you know, all of the animals together, we didn't see a really distinct response to the air puff and mostly saw a response to that withheld reward. That's what I have for my presentation. This was a really incredible experience to get to dive fully into research and be training these animals every day for this project. And I'm so thankful for getting to do this and work with you all at FYRE and work with everyone in the Celestial lab to make this happen.
Thank you so much, Leah. Okay, Chester is literally going to take us out into space. And The floor is yours.
Oh, if only they were that exciting. Not into space, but I will take you a few billion years into history. Let me get this set up.
That's pretty far.
Yeah, I think so. But not far enough for some people. Oh, okay. I also had some horrible, horrible technical difficulties with PowerPoint, actually. Regardless, I'm gonna make this work. Because this is PDF. There we go. So, if any of you saw the title of my project on the email, it was horribly long and complicated. And I thought it'd be much cuter to call it The Colors of Dark Matter, because that's exactly what we're gonna be talking about. I study theoretical physics with Graham Cribbs in the Institute for Fundamental Science. I've been working with Graham for over a year now. I'm going to continue working with him, hopefully getting a paper out on this project within the next 6 months, so to say. But before we get into the nitty-gritty, let's just talk about what the heck is dark matter. But before we have that, we have where we found it, which is not there. This is a galaxy. Since that I do theoretical physics, we oversimplify everything to a ridiculous degree, and this is actually how astrophysicists and cosmologists will represent a galaxy. And in a galaxy, there are stars, is what we denote them as. And just as we are orbiting our sun, and the Earth will go around the sun a little bit faster, actually much faster than say Pluto or Neptune would, that stars in a galaxy, we expect them in their orbits to be fast if they're close to the center and slow if they're very, very far away. And that is usually what we see when you have the stars that are orbiting the galaxy center. The galaxy center is a giant cluster of mass, typically a black hole or something like that. But there are certain galaxies we look at, we don't see that. In fact, this is what we observe. As you can tell, these stars are going very, very fast, and that's why they're very, very aggressive. And this is the famous galactic rotation curve problem that was the original evidence for the existence of dark matter. Now, one idea would be that the stars have different masses, the galaxies have different mass distributions, and people tried that over and over and over again, and none of those theories worked. Eventually, people started to think that there might be some mass there that we couldn't account for. And that was the solution. That was the origin of the dark matter paradigm, and that's the paradigm that's prevailed for past several decades. The idea is that there's this non-luminous matter, that it interacts gravitationally, as in it can affect the orbits and rotations of galaxies, but it doesn't interact with light or any other known forces, nor have we observed it in any other context. So it's dark. Now, it's time to figure out what the dark matter actually is. You add more mass and you get something that's a lot faster. So here's a very, very lightning review of particle physics. You all have heard of protons and neutrons. They are most of the mass of the universe. It's called the baryonic mass. Now, if you've ever heard of the Standard Model, it's this fella on the right. It's composed of the fermions, which includes our friends electrons, and as well as up and down quarks, which are the ones that make up protons and neutrons, as well as other similar particles to electrons called leptons, and other particles similar to the up and down quark, just other quarks. The gauge bosons carry the forces. Everybody's heard of the photon, that's for electromagnetism. The gluon is what mediates the strong force. That's what holds the up and the down quarks together into neutrons. The Higgs boson was a big whoop 10 years ago, but not important right now, so we'll ignore it. But what you really need to know is that protons and neutrons are made out of quarks and they're held together by the strong force. The reason there's 3 quarks in a neutron or a proton is because the strong force has 3 degrees of color. Instead of electromagnetism, where it's you flip one coin for positive or negative, you flip 3 coins. And so you can have a proton with a red quark, a green quark, and a blue quark, or a different particle with a red quark and an anti-red quark. Now, are these good names? That's up to interpretation, but that's what we have. Now, that's how regular matter behaves. But we don't care about regular matter right now. We want to know about the dark matter. So this is the general map of what the dark matter might be. You can really categorize it by the mass of the particle and how intensely it interacts with other stuff around it or with itself. Now, if it has a strong coupling strength, and if it's light, then it's light, there'll be a lot of it. And if it interacts very readily, then it'll form large-scale structures like we see in the universe. The electrons are an example of this. They're very, very light and they're very strongly interacting, at least on human scales. That's why we've discovered electrons, have known about them for over a century. But if the particles are super light or are super light and interact very weakly, or they interact strongly but they're too heavy, then we could discover them, but we wouldn't have already. And if they meet both of their criteria, then you have no shot. So this is the dark matter landscape, and I'm looking in this top right corner. There's very interesting searches in the bottom left corner, but the top right one is where we're looking for now. So among the strongly interacting dark matter, one reason it would have not been discovered yet is if it were also a composite particle, like the neutron or proton. So our project is all about searching for these composite dark matter particles, these neutron-like dark matter particles. And the particular question that we're asking is, how many colors, how many quarks can you fit inside a dark neutron? So here is a little drawing I made of an example of a dark neutron made out of dark quarks. We are calling dark quarks dorks because of course we have to. And they form these composite states, and they clearly become very, very complicated objects when you have many, many more colors. And colors, once again, is just the number of quarks you actually fit inside this neutron. But the central question is, how many colors could a composite dark neutron have? And would having more colors make a dark matter particle harder or easier to find? Now, the question then becomes how to constrain the number of colors for a theory of dark matter. So there are two methodologies, there are two camps of methodologies relevant to this project. It is a theoretical project, so there's no actual experimentation going on. It's purely doing math and analyzing data that already exists and creating a theory that's consistent with all known constraints. But within that, there are two main ones that we concern ourselves with. One is what's called direct detection experiments. This can set a lower bound on the mass that a dark matter particle could have. And here's a cartoon of what's the most relevant experiment for me, called Xenon 1 ton. It's a giant vat of xenon located in some remote basement in Italy, where people keep it extremely, extremely well hidden and very, very well protected. And if there's any dark matter that can go through the protection and then hit a xenon nuclei, then it'll recoil and we can detect that. And if there's a signal of there being a dark matter collision with xenon, then Nobel Prizes for everybody. Hallelujah, we found it. Hasn't happened yet, but we can use the sensitivity of this experiment and the lack of detection to constrain the properties that dark matter may or may not have. Now, the other side of it is more about the astrophysics and the cosmology. So here is a cartoon history of the entire universe. There's too much going on here. I don't understand all of it by any means, but the basic idea is that within everything from the Big Bang to now, there have been different eras and different epochs in our cosmological history. Now, what the dark matter is, its properties, and especially its thermodynamics, has a huge impact on the evolution of the remainder of the cosmos. And this is something that we can simulate. In fact, just recently, a new postdoc at Oregon, who just came here from MIT, is joining our group, and he actually published a paper that was calculating some novel effects in this kind of thermological or thermodynamic scenario. And we're going to be combining these projects, investigating his early universe simulation with our questions about how many colors could fit inside a dark matter particle. So for very tentative results, and just ignore some of the technical jargon on the right of it, these are the kinds of constraints that we have come up with for toy models, and we will be creating more robust and general constraints on parameter space for. So the blue and red regions are parts of the theory where it's ruled out. That if you say, look at the x-axis, it's the mass of the dark matter in units of tera electron volts, which is just a unit measuring relative to the energy of an electron, that if it has 1 tera electron volt, then no matter how many colors the dark matter has, that would have been detected already. And we haven't detected it, so we know that we know that particle doesn't exist. Likewise, if the dark matter is too heavy, then regardless of the number of colors, it can't exist. But there's this small window where there are feasible theories, and it is feasible to find dark matter with, say, 10 TeV in mass and a particular range for the number of colors within the theory. Now, this is a very different result than the status quo. Typically, this blue line is completely far away from the red line, that the lower bound on what the mass can be is far, far, far away from the red line on what on the maximum value that the mass of a dark matter particle could have, which is very exciting because that means we're actually able to create very predictive theories for whether or not a given dark matter candidate can exist. Now, on the flip side of that, we could be using this to make dark matter theories more appealing and essentially harder to find. Depending on your taste and how you like to play that game, then that's different ways that this project can go. But at this point, it's just a question of following our nose and continuing to come up with these models and compute the constraints that exist from existing theoretical work in simulations of the early universe and the experimental work from direct detection. And eventually, at the end of this, at the end of say my last year here, we'll hopefully have found the dark matter, and all the questions in theoretical physics will be resolved. But more practically, we'll just continue to create these constraints and try to rule out some theories of dark matter or find ones that are promising. Now, that's all I have on dark matter. Thank you for listening to this. I'd like to just thank and acknowledge SURF program for funding my project this summer. If you have any questions, please ask.
Thank you so much. Any questions for Chester or any of the other presenters? I want to take this time to thank all of you and all of the friends who attended to support each and every one of you. I am so impressed, and you did incredibly well. Yes, Professor Fauci, I absolutely agree. Great job. And enjoy the remaining last days of summer. And we look forward to staying connected. Thanks so much for spending Wednesday afternoon with us. Bye, friends. Thank you.